Hey scientists, how you going? Just want to talk you through our single-celled organisms investigation or assignment for this topic. So basically this is going to be a three-part investigation and I just want to talk you through how those parts are going to work. So it's going to involve some research and a practical. So part A is the single-celled organisms practical. So basically what you're going to have to do is to demonstrate your scientific skills to create a wet mount slide. So basically obtaining a sample and putting it on a microscope slide and using the microscope to view specimens of Euglena and Paramecium. So they are two microorganisms that exist in water. So we're going to get those samples and you guys are going to create those slides so you can look at those living microorganisms under the microscope, as well as your accuracy when creating scientific drawings and recording observations. Part B is the research component. So you're going to research a single-celled organism, choosing from either Euglena or Paramecium. So learning a bit about these. And even though this is part B, this research is likely to be done before you do the practical component. So what you need to include from your research is the name of the organism. So that includes the biological name, which is the scientific name, usually a bit longer. A description of the organism, including diagrams and labels for parts. So the diagram is something that you're going to draw yourself from your practical. When, who, how it was discovered, what it eats, how it eats, or how it obtains its energy. So every microorganism needs energy in order to live and move. Where it lives, three unusual and interesting facts about the microorganism and a reference list. So that research should be hopefully relatively straightforward and hopefully relatively interesting as well. So then presenting that research, you're going to create a presentation in a creative manner and you're not restricted by a specific mode of presentation. So most people are going to make a PowerPoint or create a poster, but you're welcome and you're encouraged to come up with something more specific. You might want to even create a video like I'm creating for you guys right now. So your due date, your teachers will go through that with you and the grade will be on your task sheet. So the first bit, or I guess the part A, is the practical. So the equipment that we're going to need is our light microscope, and hopefully you've had practice using the microscope, cover slip, cotton wool, paper towel or tissue, microscope slides and eyedropper, samples of water containing the Paramecium and Euglena. So you're going to get a little bit of, I guess, dirty water, and tweezers. So the wet mount practice, you're going to make a wet mount slide. So this is the equipment that you need. Forceps are kind of like tweezers, cotton ball slide, cover slip. So the glass microscope cover slips, dropper, distilled water, and a paper towel. So safety precautions, what are two things you need to consider? The microscope slides are made of glass, so that's something to be mindful of. That could break. It's sharp. Another thing is maybe when using the microscope, you could even write anything to do with COVID. Now with COVID, we need to use safety glasses when we're using a microscope because any virus, but that one in particular, could be transferred from a person onto the microscope and then to the next person that uses the microscope because there's that direct contact on your eye. So that's why we're needing to wear safety glasses when we're using our microscopes. So to create a wet mount slide, we gather a few strands of cotton from a cotton ball using forceps. If your specimen is too thick, then the cover slip will wobble on top of the sample like a seesaw and you will not be able to view it. So essentially you just need a few strands of the cotton wool, nothing more than that. And the reason for that is so that the specimen, the living microorganism, is actually kind of trapped and it's not going to move anywhere off the slide. Otherwise it's going to be too hard to actually see. Place 1 drop of water, don't put too much on there, directly over the specimen. If you put too much water, the cover slip will actually float, making it hard to draw the specimen. The wet specimen is the microorganism, what we're trying to look at. If you're putting too much water, it might actually float away, and too much water is messy. Place the cover slip at a 45-degree angle, so like that, with one edge touching, and let it go, and it will fall over the specimen. I like to think of putting a phone cover on your phone screen. Teacher check, show your teacher to tick it off. So this is your check. You can write your own self-evaluation comment. So the use of forceps to collect the strands of cotton wool, and then when you've done that, tick it off. So I want you to be referring to this as you go. Have you laid the cover slip down at the correct angle without air bubbles? The cover slip flat on the specimen and the slide. The correct volume of water used, 1 drop. Using paper towel to correctly mop up excess water. And the placement of the slide on the microscope correctly. So moving the stage down as far as it can go. Placement of strands directly on the center of the stage. Move the stage clips into place to secure the slide. And then correctly focusing the microscope. Put the objective lens to the lowest power. Adjust the diaphragm. Make sure the lens is correct. Turn on the microscope. Coarse wheel adjustment in order to get the sample into focus. And then fine focus afterwards. Now we're drawing our specimen using 2 magnifications. So you can do the lowest magnification at 40 times. When you draw, use a sharp pencil and draw clear lines. We're not shading or anything like that. We're just trying to draw as best as what we can see using clear lines. Second magnification, try and zoom in a little bit more. So identification of the single-celled organisms. Place 2 drops of sample water on the slide. Tease out a few fibers of cotton wool and place them on the slide. Carefully lower the cover slip onto the slide and dry off any excess water. Examine the slide using low power. Try high power. And when you're using high power, you're magnifying the image, and also that means your field of view, how much you can see, is smaller. So if the microorganism moves, it's going to look like it's moving faster, but it's not. There's just you're just zoomed in, so it's going to when it moves a little bit, it's going to look like or appear like it moves more. So sketch the diagrams of the euglena and paramecium. So 4 diagrams in total. Here is some steps to draw what you see through the microscope. So sharp lead pencil, only draw lines that you can see, don't shade or color. Draw them quite large, so 1/4 to 1/3 of a page. Record the magnification, so 40 times, 100 times, or 400 times with our school microscopes. State the name and the date of observation. Include a written description, what you can see. When the image has many cells of the same type, you only need to draw a few cells to represent the specimen. So there's the euglena and the paramecium. And then some discussion questions. Some difficulties encountered that will be unique to your experience. Propose a reason why the cotton wool was added to the slide. It's got to do with trapping the specimen. Describe the most interesting organism observed out of the 2 and the magnification. And again, that's personal preference, and that's your mark scheme here. So part B is the research. Recording the table to gather information about the organism in dot points. That's how you research, so you don't copy whole descriptions. Basically, dot points of key information. And from that, you're going to create your sentences. Don't copy and paste from websites. Do the dot points in your own words. And once you've collected the information, you're going to compile your reference list. You're going to hand this up as a formative grade or a forelearning before you start the summative presentation. So the name of your organism, get it from website 1, 2, and 3, and copy the URL into your reference list. So do that straight away. Don't forget to do that. The description of the organism, where does it live, when and who and where was it discovered, how does it obtain energy, and 3 interesting and unusual facts. The reference list, we are going to convert the website into this essentially Harvard referencing system. So what you do, the website, include the name of the organization, the date created or last updated, the date you access the website, and then the URL. And here's an example down below. So Reconciliation Australia, which is the organization, the date created or last updated, 2005 to 2009. When you accessed it, 21st of December 2009, so someone did that 11 years ago. And then the URL is what you post here. So that is a good reference there without the brackets and the EG. So you put your 3 references there. Okay, this is going to be what your teacher gives back to you before you begin your presentation. So basically you'll get given an A to E grade for the detailed notes for the questions, correct answers for those questions, and then citations, so you're referencing. The presentation, You're not restricted to a specific mode. So the guidelines, colorful and appealing appearance, including pictures. So you have to have some sort of visual component to your presentation. You can't just talk without showing any pictures. All sections from research must be covered. The sections are clearly defined. So e.g. the description of the organism is separate from where it lives. So if you're doing a PowerPoint slide, have a separate slide for each one. Creative mode of presentation. You could use different websites. You could make a website. You could do Prezi. There's lots of different aspects or things you can do. You can make a video. Your own drawing of the microorganism is seen under the microscope slide. So basically take pictures and upload those pictures or screenshot them from your assignment and put them in your presentation. And this is a self checklist for you. Okay, and then this is going to be your of learning presentation. And you can see it is about your presentation 100%. The other two parts were leading up to that. Thanks guys, good luck with your presentation.